Hello, welcome. This is Mr. Watkins. This is part 2 of the protein synthesis lecture. And so we just want to recap. Transcription occurred inside the nucleus. This is the nuclear envelope, so the nucleus would go all the way around here. And so we had that copy made, and that copy is messenger RNA. And the messenger RNA comes out to the cytoplasm, and it basically hooks up or gets stuck with or gets put into a ribosome. And I'm going to write some stuff here to kind of give you an idea of what I'm talking about. The ribosome has two main parts. The two parts of a ribosome, in fact, let me get another piece of paper, we'll just draw it out. The two parts of a ribosome are, well, I guess I have to stick on this one. The two parts of the ribosome are a small subunit and a large subunit. Now, typically it's drawn in this fashion. There's the small subunit and then the large subunit. And I didn't quite draw it to scale, but the idea being that this mRNA in the cytoplasm will work its way right between the large and the small subunit. And as it goes through, it gets read in a particular frame. So I'm just going to hold this piece down here. I'm going to probably put a little piece of tape on it so that we can kind of see it and it will stay put, because we're going to have to mess with it. And so this piece, if you'll just imagine that a large subunit or a small subunit has attached to it, and then that the large subunit has attached to it here, and we're reading, now we're going to be reading this, this strand to make a protein. So you're going to need to get your codon chart to do this part, this next part. So here's one of the codon charts. You can download this off of the internet. There's just a ton of them out there. This is the one that we use here at school. And what we're looking for is a very special sequence, a start sequence. What happens is that this ribosome is looking for this very special sequence, and that sequence on the mRNA is A, U, and G. And if you look carefully, you could see that sequence here. So we got ACA, and it's looking for it in groups of 3, and I'll explain that in a minute. Then we have CAU, no, that's not it, AUG. So here, right here, is that sequence that we're looking for, that sequence of 3. And this sequence here is the code that tells this ribosome, hey, we need to start making this protein. Now, out in the cytoplasm, floating around near the ribosome, are these smaller pieces that are ribosomes. It's part of our ribosome list. And these small pieces are tRNA. If you'll remember from the first video, the tRNA is called transfer RNA. It does the actual translation. The ribosome does the reading of the message, the mRNA, but the tRNA actually does the translation. And so what ends up happening is this tRNA floating out here into the cytoplasm. It only has 3 bases on it. I need to make sure I can line up my bases here real quick, so just bear with me. It's got 3 bases on it, and I'm going to put those 3 bases on this end. And then on the other end, it's got an amino acid, say like methionine, and we'll just abbreviate MET. So here we have our tRNA. This is our tRNA. It's going to have a 3 base sequence on it. It only has 3, and that sequence is going to end up being the complement to this sequence here. So we'd end up having U, A, C, C. And so when this piece attaches, the ribosome has found its 3 AUG pieces. This piece of tRNA is going to come right on in, and because these are the complements, they're going to attach, and this tRNA and stuff is going to sit right here and bond, and the whole thing will then shift down 3. Now we need to talk about a few things before we go any further. This 3 base sequence right here, this piece right here, is called a codon. A codon is a piece of the mRNA. It's 3 bases, and it is basically the reading frame by which the ribosome basically knows, hey, this 3 bases here, we're going to have a 3-base tRNA that lines up to it. And so when you look at your amino acid codon chart, you're going to look and try and find AUG. And on the codon chart, it's probably hard to see because it's so small here, but on the codon chart, AUG is the code for methionine, and that is our start amino acid. This is our start codon. Okay, so here we go, and we have this that's attached, and we're good to go. When this whole thing shifts, it doesn't shift down 1, it shifts down 3 bases. So we're going to shift down to UUG. So let me grab another pencil here, and we can just mark it just so you can see the difference. This is still a reading frame. This is still the ribosome reading it, but it's reading it in groups of 3. And so out in the cytoplasm, somewhere out here in the cytoplasm, there is a 3-base structure that is, or 3-base tRNA that lines up with that particular amino acid sequence. So let me cut off another piece of paper here so we can start putting in these sequences. So we've got this 3 bases here. So there's going to be a tRNA that's floating out here in the cytoplasm. This tRNA is going to have an amino acid attached to it. That amino acid that's attached here is the amino acid that we would see if we were to look up UUG. So we've got UUG here, so that's leucine. So we're going to have leucine here. And then we'll have the complement on our tRNA, so this is going to be adenine, adenine, cytosine. So this thing was out floating in the cytoplasm, and when the ribosome moved down this reading frame and saw that it was UUG, this piece of tRNA was able to come in and bond, right here with this particular piece, with this particular codon. Now on the tRNA, on this piece of tRNA here, this 3-base, this triplet, if you will, is called the anticodon. It is that triplet, that same triplet, that is the complement to the codon. And on that tRNA, it's going to carry that other amino acid. And then once again, we're going to shift down 3 bases. The whole reading frame will shift. This ribosome, which has the reading frame on it, the whole ribosome will shift down, and we're looking for a piece of tRNA that's going to have GAC on it. And it will have an amino acid out here on the end. To know what amino acid is, we have to read the message, not the translator. So we have to go back to our amino acid, mRNA, codon chart, and we're looking for CUG. So we're looking for CUG, that's also leucine. Now here's an interesting thing to point out. There are 20 amino acids. These 20 amino acids, you've got a bunch of different possible combinations of these codons, and so some of the codons, even though they may look a little different, or they may be even actually quite similar, they may code for the same amino acid, and we'll talk about mutations in a different video. Now once this thing attaches, it's going to form a bond between those amino acids. I mean, they are so close that we're going to form this bond here between these amino acids. What ends up happening here is that same condensation reaction we discussed, or dehydration synthesis reaction that we discussed previously. Water will be formed, and this bond, because it's a bond between amino acids, will be called a peptide bond. So once again, this thing's going to shift down. Now this time, when the thing shifts down, this part of the tRNA right here, this is going to go back into the cytoplasm. It's going to go back out and find another methionine amino acid and be charged with it. In other words, have that attached to it. The whole thing is shifting down again. We have our next set of, our next codon. There should be a tRNA molecule out in the cytoplasm that will come in and attach, and that tRNA is going to have that anticodon on it of CUU, And CUU is that leucine again? It is leucine. So we've got leucine here again. Oh, excuse me. I just made one of the most classic mistakes that my students make. I read the tRNA, and it's not the tRNA that you read, it is the mRNA that you read. Now, I know that may seem kind of cheesy because I did set it up this way, but it's really, really, really important that when you read the message that you're reading the mRNA strand and not the tRNA strand. So we're actually looking for GAA. So GAA, that's going to be glutamic acid. So don't make the same mistake that I just made. It's a very, very common mistake, and I see it all the time on tests. Remember that you are reading the mRNA and not the tRNA here. Again, we're going to have a bond form. That bond is called a peptide bond. The reaction that does that is condensation or dehydration synthesis. Again, the ribosome is going to shift down. It's going to continue to shift until it gets the 3-base combination that says stop. There's not going to be a tRNA that goes with it. So let's see if this is it. Now remember, we're not going to read that tRNA. We're going to read the mRNA chart. And what we're looking for is UA and G. So there's UA and G, and that does mean stop. Now in pre-AP bio, we don't get into any more detail than this, but in AP and IB, they will talk more. And when you take that class, one of those classes, you will talk more about what really happens here, but there's actually a special factor that comes in and releases it. Again, I want to emphasize that as these peptide bonds are made and as this shifts down, eventually all of these tRNAs come off. And so that all we're left with is the methionine, leucine, and glutamic acid all here to go on to the Golgi apparatus. So the amino acids, now this protein, this amino acid sequence, because the tRNA is now gone. In fact, I'll just make it easy on both of us and take off those tRNAs real quick. The amino acid sequence has been made, is now gone, and this piece is now going to go probably to the Golgi apparatus or the Golgi body so that it can be repackaged, maybe folded differently, put with other proteins so that it can then get into a vacuole and then go to its destination where it belongs. Remember, transcription occurs inside the nucleus. Translation occurs inside the cytoplasm. Translation uses mRNA, so does transcription. Translation only uses tRNA, only found out into the cytoplasm here around the ribosome. The ribosome is what's going to read it. It's going to set that 3 base pair, that triplet, that codon to read, and to read the sequence on the mRNA. You'll need to use your mRNA chart, which I have right here, and that will be given to you on your exam. Even the AP and IB exams, they don't expect you to know those charts to tell you what that amino acid sequence is. Once the amino acid sequence is put together, remember, peptide bonds will form between those amino acids as it's being read. The tRNA that's attached to that amino acid will go back out in the cytoplasm. But once that's all read and we've got our stop and it says stop and the amino acid, now a protein, has been removed, it will go to that Golgi to be repackaged and then distributed in the place where it needs to be. That's it. Thank you.